All right, everyone. I'm sorry for the delay there. Welcome, though, to the Astrophysics McGill public live stream for this month. This evening, we have Dr. Miguel Fernandez Martinez giving us a lecture about what extreme microorganisms teach us about where life might be elsewhere in the solar system. So he'll talk for roughly half our session today, which goes to 8 p.m., and then you can ask him your questions. So if you're watching along on Zoom, please type your questions into the chat. This evening, we're doing the live stream over YouTube, so you can type questions into the comment on the YouTube channel if you're following along there, or in the comments on where we've shared the YouTube link on Facebook. Just send them in, and we will make sure they get to Miguel at the end of the night. So, without any further ado, Miguel, please share your screen and take it away.
Okay, thanks everyone for being here tonight. Really happy to have the opportunity to talk to you today. I want to apologize for my English, so sorry about my accent and my grammar errors. I will try my best, but nobody knows what happens on a Thursday evening. So far the week has been hard, so I'll try to do my best, as I said. Okay, yeah, as Leah said, I'm going to talk to you about what streamophilic microorganisms can teach us about looking for life in the solar system or everywhere else in the universe. The title of the talk today is My Bacteria Teacher. I don't know if it recalls you anything, but I decided to use the name similar to this one from the documentary that won the last Academy Awards this year. My Octopus Teacher. I really recommend you to see it, to watch it, because it's quite interesting, quite fun. And it talks about two things that could be applied to this talk tonight. First of all, well, I don't know if you have ever think that, but it's something that comes around my mind every time I see an octopus. It's like they look totally like aliens. Here you have a picture of it, so you imagine an alien being in some kind of form. For me, it will look like an octopus. And also, apart from that, yeah, the film talks about how this man, the man who recorded the documentary, finds something really interesting just above the corner, just around the corner, and on a beach that is really close to his place in. I don't remember. I think it was in South Africa. I don't remember if it was in South Africa or in Australia. Sorry about that. Well, yeah. So it's a kind of thing that I want you to feel this evening, hopefully. So you will find some things that are very close to us, but are related to incredible things that we have thought many times, like what's the origin of life. It's life present anywhere else in the universe? Are we alone? That's the question that you could see on the graph that Astro McGill shared into the website or in the social media. Well, that's my goal today, just to engage you into astrobiology and also have a good time. So let's go. Okay, so yeah, I work for the White Lab that is focused on astrobiology and polar microbiology. It's here in McGill University, part of the Natural Resource Sciences Department. And well, I guess you are familiar about what polar microbiology means, but probably you are not so familiar with what astrobiology means. So first thing that I want you to show today is a good definition for astrobiology. So astrobiology can be defined as the science that studies the origin, evolution, distribution and future of life in the universe. It's a broad definition, but I think it's a very accurate one. But I don't know about yourselves, but about me, reading this definition, another question comes to mind is, what's life? we know what's a living organism, but if you have to define in, I don't know, 15 words or something like that, what life is, it's a very difficult thing. So I used to, as we are going to talk about the space and space mission, I like to use the definition that NASA give us. Life could be defined as a self-sustaining chemical system capable of Darwinian evolution. So yeah, we know how, as I said before, we know how a living organism look like on Earth, but following this definition kind of bring us to the question of how can we imagine the living organisms can be in any other place in the universe? So probably most of you, as me, for example, would like to think that a living organism in other parts of the universe would look like this one. It would very cute. It would be very cute. It would be very funny also. But probably following the definition that talks about chemical self-sustaining system or whatever, they will probably look more like these things here, like microbes. And yeah, these definitions and thinking about life brings me to the point of thinking on other questions. So I don't know if you feel the same, but I kind of wonder if there's only one single origin for life in the universe. That could mean that probably we are very similar to each other, and here you have me wondering about that in a starry night. If there were only a single origin for life, it's possible that we could find life in any other place in the universe. And a third question that came to my mind, it's if finally life is present in other places in the universe, are we ready technically to detect it, to know that the organisms that are there are so similar, or at least similar in some way that could lead us to say that they are living organisms? So yeah, there's so many questions that came to my mind. These are only 3 of them that I will try to answer through this talk today. Talking about first, that it's not the main topic of the talk, but I want just to share you some things. There was only a one single origin for life. Well, maybe some of you have ever seen or read about these phylogenetic trees that shows that every organism on Earth has the same antecessor. His name is, or her name will be Luca, Luca, that is the last unique common antecessor. But well, we are not talking about that. We are talking about something that came before that. This is a study that was published this week. I think it was published on Monday on PNAS journal. Very good one for those of you who are not familiar with scientific journals. And they talk about that they discovered this compound, ethanolamine. Why is this important for us today? Well, this ethanolamine is the compound, one of the basic compounds of the cell membranes. The one that is in the head of the phospholipids, the one that is here. Here you can see how a cell will look like. Here is the membrane. Here's the structure of the membrane. And on the head of this bilayer, this phospholipid bilayer, we have this ethanolamine. They discovered this, and not in a cell, of course. They discovered this just in the middle of the Milky Way. They discovered it in the, let me look for the actual name of it, in the interstellar space and in meteorites. So finding this molecule that it's so important to create these membranes and to enclose the chemical system that we have mentioned before in the middle of the Milky Way, it could mean that this molecule that is so important can be widespread on the universe, or at least in our galaxy. So if these molecules are so important for creating cells, that is let's say the minimal unit for living organisms, could lead us to think that, well, if it has arrived on Earth, it has arrived to Earth, why it's not present in other places in the world, in the universe, sorry. And that leads me to the second question. If the origin of life was only one, why, or it's possible to find it anywhere else in the universe? And it brings me the idea of what life needs to what kind of environment life needs to have to be able to develop. And it brings me another definition that I want to share you today. Is the definition of habitability. Oh, it's a very difficult word to say it in English. Sorry about that. So it can be defined also as a kind of measure of the planets or any other natural satellites that those planets can have potential to develop and maintain environments hospitable to life. That means that life can be kind of I don't know, that life can like these environments just to develop, not to only be originated, but also to stay and develop and evolve. Well, you could think, what are these environments? What are the limits of these environments? What are the minimum units of habitability that life needs to develop? Well, you can think on your own, and you can think on the human beings, and especially these times of the pandemic, you will realize that having a Wi-Fi connection could be a sign of not habitability in any other planet than Earth. So probably for us, it's going to be very difficult just to find a place that we can call home anywhere else in the universe, at least at this moment. I don't know how it's the Wi-Fi connections on Pluto, but on the rest of the planets, it's not really good. But we're not talking about human beings. We are talking about microbes. We said before that this is probably the let's say the most possible organism that could have thrived through these conditions in the universe. So for them, they only will need something like this, very simple environment. Indeed, they will need just three different elements. Let's call them three different parts to be habitable. First of all, they need six different chemical compounds. The so-called CHOMPs: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These are the basic elements for every living organism that we already know. Apart from them, you see that they are present here, they will need an energy source. An energy source, it's nothing but a chemical name that we give to a pair of molecules, one of them acting as an electron donor and the other one acting as an electron acceptor. So probably they will work like this, like having a little amount of iron and, for example, ammonia that you have here, and they could just be transferring electron from one to the other, and the organisms will make the most of this transference of electrons and transforming it into energy. And what's the final thing, the final element to have in a habitable environment? Water or some other kind of solution that could that permits chemical reactions to take place and to act as a buffer between different substances, different conditions of the environment. But well, you see that there are only three different things that we need. And why don't we think that this is something that could happen in the universe, or could have happened before in the universe? We are not talking about organisms that are living today, but probably organisms that have lived before. That brings me also another thing to think about. Very important thing. Habitable does not mean inhabited. I mean, being an environment that harbors all these things, the compounds, the chemical compounds, the energy source, and the water, presents the minimum things to be habitable, but not. It doesn't have to be inhabited by itself. I mean, having the condition doesn't mean that it's going to happen. That's what the NASA missions are doing today on Mars. Here we have Curiosity, and here we have Perseverance and Ingenuity. And what they are doing is just to look for habitable environments. And after finding them on Mars, as I said before, it doesn't have to be habitable now, but probably habitable in the past. After finding those habitable environments, they are trying to find any sign of life on those environments. Not an extant life, that means that an organism that is alive now, but something that tells us that there before had been some kind of living organisms. So that's what they are working right now on Mars, on Mars surface. Curiosity is more intended to find these habitable things, and Perseverance is also looking for these signs of life. And future mission, as the one, for example, from the ESA, the European Space Agency, Mars 2022, they are going to look for real signs of life, probably better than Perseverance. So yeah, that's a very important thing. They have state-of-the-art instrumentation, but they are some kind of limited in comparison of what we can do here on Earth. So until we have something like this, something that could let us travel through the Mars surface and find and do experiments ourselves. And I think Elon Musk must be working on something like this. We have to go to other places closer to us that shows these kind of conditions that resemble in some kind of way the condition that we have in other planets, especially we are talking about Mars today in this moment. So that's what we named as terrestrial analogs. As I said, terrestrial analogs are places on Earth, so close to us, that following one or more of their environmental parameters resemble other solar system bodies. It could be Mars, it could be some kind of moons. I'm going to talk about them later. And yeah, we have a bunch of them. They are not so strange. That's why I told you before in the introduction that I hope that you are going to find that round the corner you will find, well, maybe not round the corner, but not so far, you will find interesting things as we do here in astrobiology. So one of these environments that resemble Mars in some way is the Antarctica. There are some, let's say, not a lot, but many environments within Antarctica that resemble Mars. They resemble Mars in terms of temperature. They resemble Mars in terms of UVA radiation. For example, these mountains here that are called nunataks is a Inuit word that means a peak of a mountain range that protrudes, that gets above the glacial or the glacier ice or something similar to this. And we have just published, well, not published, but a paper, a scientific paper has been just accepted talking about this, the strategies that the microbes here follow to thrive and to be alive in this under these conditions. They're very interesting. They try to hide from the UV radiation, but also try to find the places where the nutrients can be trapped, and also maybe, let's say that they are very adapted to the conditions, but depending on the substrate that you look at, well, doesn't matter. It's a very interesting thing, and we have found that they are the microbes there are teaching us how microbes on similar situations on Mars can have thrived. Another environment close to this one, indeed, it's in the same island, in the South Shetland Islands, is in southern South America, let's say that. It's something like this, where you have the glacier front here with a lot of ice and also different layers of volcanic ashes. They come from a close island that is a volcano, an active volcano in Antarctica. And it also resemble different places on Mars that could have had this combination of water ice and also volcanic acids with all the components that they will need to thrive. Remember that we need the source of energy, we need the compounds, organic compounds that could come with the acids, and we need water that is in the ice. So especially these two environments resemble what happened in an era on Mars that we call early Mars. So as you may know, Mars once in a time at the beginning of the well, let's say at the beginning, when the planet started to be created, let's say that at some point it started to harbor to have a lot of water, a big ocean in the north hemisphere. But after it started to lose their atmosphere, it started to lose also the liquid water on the surface, because of the freezing temperature that it was facing. So Mars changed from this situation here with liquid water on the surface to something similar to this one, with just a couple of places on the planet with water ice on them. So as I said before, Antarctica and those environments, especially you may recall the nunataks that probably are like these spots here, like mountains that protrude over a totally icy surface, like glaciers or something, are very similar to this. So we are learning a lot of things about how life could have thriven on Mars if ever did. Another environment on Antarctica that resembles the situation that we have today is the dry valleys. These are far from the place that I have showed before. They are closer to the southern part of South Africa, if I am not wrong. And as I said, they are called the dry valleys. It's not a name that came from imaginary or whatever. It's a real thing. They're really dry, they're really cold. If I am not wrong, I kind of remember that they only have, I think it was less than 3 days of available water for the microorganisms to thrive. Only 3 days a year, and less than 3 days a year. And also they saw very high UV radiation, and they also saw very low temperatures during the year, like around minus 20 Celsius or something like that. So very, 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 very cold. As you can see, they are very similar to what we can see on Mars today. They are really, really similar to this. And what happens on this environment that looks to be devoid of life? Is there anything alive there, as we can think that it could be on Mars? It is. What we find there is something similar to this. This is a rock collected into the dry valleys. And this is the strategy that the microorganisms follow to develop on these environments. They are within the rocks. They are called endoliths. That means inside the rock. This is the rock surface. And this green layer here, it's a layer of photosynthetic active microorganisms that are here not for only one reason, but for many of them. They are protected from UV radiation that will come this way from the surface of the area. They are protected through this couple of millimeters here. They are also protected from the colder conditions that can be surrounding the rock in the environment. Here on the rocks, the temperature can be even more than 20 degrees above the temperature on the surface, on the soil or whatever. And also these rocks, especially these ones that are called sandstones, you can see the grains of sand here put together, they're very soft. These rocks are very soft. They also act as a trap for water because of their texture, because of their minerals. So here they have all the things that they need. As they are also photosynthetic organisms, they can just create their own organic matter. And they have the water better than on the surface, and they have the protection from the UV radiation. So they have everything they need to thrive, and they thrive, as you can see here. This environment, as I told you, is very similar to what one can find on Mars today, but they are not totally similar to these environments on Mars. So what we are doing here, we already know that these microorganisms are here, but we are trying to discover what would happen to these microorganisms that that are in this very, very harsh environment, if they are subjected to the real condition that they will face on Mars. And that's what we are doing now. We introduce these rocks in this chamber here. This is chamber, this chamber is called Marte, that means Mars in Spanish, is in a Spanish institute. And what we have done, especially one of my colleagues, I don't know if she's present today here watching us, Catherine Maggiori, it's her project. We have introduced the rocks here in the chamber, and here in this chamber, you can control all the conditions, all the environmental conditions that you can have on Mars. You can control UV radiation with these lamps here. You can control temperature. You can control humidity. So you can just have the samples during whatever you want. Let's say a week. We had them inside for a week. Here you see how it looks like when the chamber is working from outside, of course. We put them there in this chamber for as I said, a week with a very low temperature. I think it was like at -20. No humidity in the atmosphere, and with a complete UVC radiation firing them, try to fry these microorganisms inside. And we are trying to analyze the data. Is what we are doing. One of the things that we are doing in these days. We have found that microbes probably are not alive anymore, but we are still able to find and to recognize different biosignatures, different biomarkers, different marks, as I said, that Perseverance is doing on Mars surface, trying to find some chemical compounds that tell us that they belong from living organisms before. We are trying to figure it out. So you probably will have news in, let's say, less than 6 months, hopefully. But it's a very interesting thing that we are doing. I wanted to share it with you. Other environments that are similar to Mars and similar to other solar system bodies, to other bodies in the solar system, are even closer to us. They are in the Canadian High Arctic. They are in Nunavut, in the Axel Heiber Island. It's called. It's very far north, close to the North Pole. I think it's the North Pole is in latitude 90 degrees, and this one is latitude 80 degrees, something like that. And what you have here is this spring that presents liquid water all over the year. Why it's possible to have this water here? Because the water inside it's very salty, very, very, very salty. It also presents high concentration of methane, and it also presents high concentration of sulfate. So it's kind of a toxic water for us, but not for microbes. Indeed, we have been studying this. It's another study that we are carrying out in our lab. In this case, the first author is Elise Magnusson. And we have found that a complete microbial community is active there. Do you remember the electron donor, electron acceptor that I mentioned before? Well, here they are using these compounds with sulfur, this is methane, this ammonia here, just to transfer the electrons from one to the other and obtain energy from that. And they are also thriving in the conditions, in the cold conditions that can be there, especially in winter, and also the conditions of the aerobic or anaerobic condition, that means with oxygen or without oxygen. So yes, and if you have a look at this environment here, you will also see the similarities with other solar system bodies. We are not talking about Mars. It can be also be recognized as a Mars, early Mars analog, because one of the theories that said that liquid water persists longer on Mars surface after the big oceans disappeared is having this kind of brines things, like salty things. So with water, with a lot of salt, you know that the fusion point decrease, so you need less temperature to have this water liquid. And it's another explanation that they have in mind, just to explain or to try to look for biosignatures in other places on Mars where liquid water persists longer. But apart from Mars, they also resemble. Just have a look at them. Have this image in your brain for a second, and then compare it to this one. This one is Europa, is one of the moons in Jupiter. At least for me, I don't know for you, but the environment looks very similar to this, like ice, a lot of ice, and these kind of different color things. Could be salts, could be methane, salt methane, I don't know. But seems very similar to me to the high Arctic. I didn't mention the name, but the name of this spring is Lost Hammer. Also, the name is very cool. Not only the microbial communities that you can find there, but it's the name of the place. It's cool. Yeah, that's a similar situation that you can find here in Europa. And in Europa, it's very interesting because they have been said that there are some kind of subsurface oceans that can let's say have this upwelling thing, upwelling water and upwelling nutrients through cracks in the surface. So very similar to what we have seen in Lost Hammer. It's like water flowing below the surface, probably below the permafrost, this layer of the soil that is permanently frozen, and go to the surface in these cracks. So very similar. It's like a geyser-like places. In this case, they can be very similar. If this is really happening in our place and in Europa, they are very similar. And as we have seen, the compounds on the water allows the microbes to develop. So another place that there is very similar to this is Enceladus, a moon in Saturn that also it's thought to have these below surface oceans. And also, apart from these two moons, they are called the icy moons in the astrobiology world because of this temperature that they have and this ice that they have in the surface. The Lost Hammer spring also resemble situation that could be found on Titan. Titan is the biggest moon in Saturn, and in this case, it's not similar to Lost Hammer because of the ice, but it's similar because of the methane that they have in the surface. It's believed that there are many lakes that are very, very highly concentrated on methane in the surface. So as we have said before, if we have a similar situation on Earth that presents living organisms there, why are not going to be the same if life, let's go back to the start of the talk. If life started at the same time, it has only one origin, why it's not going to be present in a similar way as we have seen here on Earth, but on these moons? Well, something to think about. We are getting steps farther, but we are very far yet just to know this. But yes, bear in mind these kind of things. Yep. Apart from that, there are also other places close to us. This one is an image of the front cover of Astrobiology journal of I think it's the actual issue, the issue that just came out this month on May. And this picture comes from a paper that we published. In this case, the main author was Brady O'Connor. He's a PhD student in our lab. And he studied and he found out that in these caves that are formed by lava, they are volcanic caves. Again, this could be present, this kind of caves could be present into Mars. I think that they have already seen some of them on Mars. They have inside this dark thing that you see here. It's not water, but it's ice. So it's something that is very cold inside. And what Brady and our group find out is that inside this ice, in this Mars analog environment, there are a lot of microorganisms living and being active here in the ice. They are very similar to cold adapted microorganisms on surface. But here, remember that we are beneath the surface, and on Mars, it's very important to be protected, especially not only from the extreme temperatures on the surface, but also from the UV radiation. Remember the chamber when we mentioned the UVC radiation, that is the most damaging one for microbes or for life, to be honest. And here is where they can find this kind of protection. So yeah, this is one of the best examples and one of the first papers showing that we can find microbes here in the caves being active. Apart from cold places, we can also go to different places that are also terrestrial analogs. This one here is Rio Tinto. I don't know if you have ever heard about that. The planetarium here in Montreal has the name because of this place. It's in southern Spain, in my country. It's a very impressive place. This is an actual river. So what you see here is the water of the river. It's totally red because of the high amounts of iron that it has on the water, and also the high concentration of other metals, of other heavy metals that are here. And even with this, and it's also one of the most acidic places in the world, at the beginning of the river, you can find pH values of less than 1. I don't know if you're familiar with pH, probably you are. pH is a scale that measures the acidity of a substance and goes from 0 to 14. 7 is the limit, the neutral pH, neutral acidity. Below 7 is acidic, and over 7 is basic. So here we have 0.5, something like that. Imagine it's like the limit of the pH here. And even with these conditions, there are a lot of microbes. There are a very high number of microbial mats, like this one that you see here, red. And also you can see algae here, green algae. So they are adapted to these conditions. This is also interesting because, as you may know, Mars is called a red planet because of the iron and other compounds that it has in the surface. So again, if water was present on Mars and it has this acidity, this concentration on iron and other compounds that are, one can think that they are not compatible with life. Here on Earth, we have seen that they are compatible with some kind of special life. And also, let's talk about the last one, the last terrestrial analog that I want to share with you. That is the Atacama Desert. The Atacama Desert is known as the most ancient and most the driest desert on Earth. As we said, it's similar. The conditions are really similar to what one could find on Mars. Again, if you have a look at the Mars surface, they are very, very similar to each other. And also on the Atacama Desert, we have found life. Maybe not in the surface, but within rocks, as I showed you before on the rocks from the Antarctica, and also beneath the surface. We are now also having another project studying the surface the subsurface of the desert, where we have found different places that presents a lot of water, especially a lot of water if you compare them with the surface. So yeah, we are trying to see if the microbes there are alive, they can thrive or not. Again, see the picture, they're very, very similar. Yep, that leads me to the last question. The last question that I was wondering in my picture with this magnificent night. If life has an origin, a similar origin, or only a single one on the universe, and we have found out that there are some habitable places where this life can thrive. Remember, being habitable doesn't mean that it's inhabited. Are we going to be allowed to recognize this kind of living organisms? Well, stay calm, take it easy, because we are working in the White Lab Polar Microbiology and Astrobiology Lab. We are working on a totally automated platform that is called Microlife Detection Platform, that has a lot of instruments inside, from taking samples to analyzing them, to see if we are going to be able to automatically, let's say robotically, or whatever you want, we are going to be able to recognize this life, these kind of these living organisms in the future. So this Microlife detection platform is being developed right now, but we are hoping that we are going to send it to space in some time in the future. So stay tuned, because probably in the next missions of the CSA, NASA, ESA, you are going to hear about us again hopefully in not a long time in the future. And that's all I wanted to tell you today. I hope I wasn't too long. Well, it's been almost 40 minutes. If you have further questions, or you want to contact me or contact whoever in our lab, here you have my email account. And also you can find me within the social media. I have a YouTube channel, I have a Twitter account, and I have an Instagram account. with this name, científico. It's a Spanish name. I'm sorry, most of the things that I post on this social media are in Spanish, but we can always make a translation for you. You have to stand with my English level, but I think we are going to be okay. Yep, that's everything. Hope you have fun, and thank you very much.
Wonderful. Thank you so much, Miguel. That was great. We have tons of questions coming in already, so if it's all right with you, we'll just start giving you some of them.
Okay, I stopped sharing my screen. I don't know if you want me to continue sharing it or not.
It's your choice, right?
Okay.
Yeah. So first off, could liquid methane replace water as a medium to allow for the chemical reactions required for life?
That's a very good question and a very difficult question to answer for me. Yeah, I'm a microbial ecologist. I'm not a chemist. As far as I know, methane can be liquid, but I think it's very reactive. That means that probably you're not going to have the same reaction as you will expect to have in water, because water is kind of an inert thing. It's very difficult to use the atoms within the molecule of water to be used for a different thing. So as I said, I'm not an expert. I don't know almost anything about that, but I would say that it's very difficult to have it replacing water. At least if the lakes, I think that maybe this question came after talking about Titan. The lakes in Titan, if the methane is highly concentrated, but not the only compound on the lakes, it will have, from a point of view, will have a greater opportunity just to bear life or to replace water, but not by itself only.
Okay, thank you. Kind of in the same vein, apart from the set of elements mentioned, like iron, carbon, nitrogen, etc., is it possible that another combination of elements not seen on Earth could produce life?
Yeah, that's a very good question. Also, yeah, there has been some experiments trying to replace. I think it was replacing carbon, that is like the main element that you need for organic matter. Replacing carbon with, I think it was silica. I don't remember. And the problem is that, as for methane, methane is carbon and 4 atoms of hydrogen. It's very reactive, and there's no, as far as I know, there's no any other element that can act as carbon. So it's the opposite reason. Carbon have this special, let's say, special situation of its electrons that permits to have a very reactive reaction with a lot of different elements that are needed for life, as we know on Earth. Probably that's one of my fears. That's why I told you to stay calm on with the last slide, because yeah, probably we are going to find life that has nothing to do with life on Earth. It's probably they are based on a totally different compounds, a totally different chemical composition. But well, there's this thing that is I think it's the Occam knife. Let me correct me if I am wrong. That basically says that if something is very easy to understand, it's very easy to happen, why it's not going to happen? So we have already known that life has appeared on Earth. We know how it looks like. Yeah, Occam's razor, thanks. So if life is like that on Earth, why it's not going to happen the same way in other planets, in other solar system bodies at least? So that's why we are always looking for something similar.
Mm-hmm. That makes sense, thanks. Do you think that life could exist on Europa given the high levels of radiation there?
Yeah, because remember that Europa it's supposed to have these subsurface oceans. So Europa has the protection, the subsurface ocean has the protection of the above ice layer. So probably we are not going to find, or we are finding biomarkers, but not living organisms on the surface. But we can find some biomarkers coming from these upwelling things, these upwelling places, like geysers or whatever, as I said. So probably they are going to be alive beneath the surface. I'm not sure if they are going to be alive in the surface.
Okay, cool. Do you know any sampling techniques used for extremophiles located in subsurface and subglacial environments?
I exactly don't know what this person is asking, but yeah, you have this for sampling directly. You have these drills that goes below the surface, and you can just get sections of the different depths. So I don't know if he or she is talking about this. It's like not sampling directly them because you have to take them out, and yeah, it's difficult just not to get the sample contaminated with above material or something like that. But yeah, what we normally use is these drills that can be sterilized, and you enter them into the surface, take the whole thing as once, and then you know where the section belongs to, and say, okay, these are from the subsurface, these are closer to the surface. This is the way that we do.
Okay, thank you. There was talk of a means of burrowing beneath exceptionally thick icy moon surfaces. Is that still on the table? And if so, will it happen within our lifetime?
Yeah, that's another good question relating to maybe what we can say is science fiction. I don't know. I don't know, because you know that technology advances really fast. So I don't know what's going to happen in next 20 years. I think that it could be easier. And I think it's a proposal of a project called Dragonfly. If I'm not wrong, I cannot recall where this mission is going to. I think it's in Enceladus or Europa, but I'm not sure. Maybe it's Titan or another one. Yeah, this is like having this drone flying over the surface and finding these upwelling things and just taking samples from the water or the gas that came from these upwelling things, the geysers. I'm saying a lot of time geyser today, but yeah, it's like coming from them, because it's gonna be easier just to sample something that it's coming from beneath the surface, that instead going below the surface to find it. You have to land the thing on the moon. You have to you need to have this drill, but very big one, because you don't know how big, or at least I don't know how deep is that this crust. So I think something like that will be the next project, and instead of being drilling there, just having these dragonfly things or similar. Yeah, I think that someone in the chat said that dragonflies for Titan. So thank you. Yeah, I couldn't remember, but maybe you can have this same approach for the other moons.
Wonderful, thanks. How much value do you place on the searches that you've been describing here versus those searches for like faraway signals, like radio signals using telescope?
Ooh. Yeah, well, I know nothing about these kind of things. I know almost nothing about astrophysics. But yeah, well, what I can say is that telescopes and these kind of measurements, as the one that I mentioned, talking about this ethanolamine that has been found, allows biochemists and allows biologists just to make their hypotheses. I don't know if we can just, as a biologist, we can just make an hypothesis based only on that. I think that they are more intended to give us information about the existence of planets in the habitable zone. That is another thing that I haven't talked about, but within a solar system, you will have this habitable zone that where the energy coming from the star is enough to keep these planets or these bodies hot enough, and having this energy coming from the sun in enough amount or whatever. But I don't know if they can be used, at least now, for something farther than having these planets or these things. Ethanolamine probably it's an example of that, but I'm sorry, I'm not the right person to answer that.
not at all. There have been in recent times certain asteroid samples returned from space. So the question asked here lists one's from Japan, one from China, some from Russia, and what has man learned from these past missions?
You mean that the missions that has been landing on the or going?
I guess asteroid return missions.
Yeah. Well,
return.
yeah, I haven't read the results from these missions, but probably they will give us some information. I'm going back again to this latest paper from this Monday. They compare the amounts of ethanolamine on this intergalactic space with the ethanolamine found on some meteorites, some asteroids. So they say that it's pretty similar, the same. It's pretty same. So what we can learn about that is probably it's a theory called Panspermia. Maybe the person who asked this question known about that. It's something that says that the life on Earth wasn't originated on Earth, but came from another place on the solar system or on the galaxy or on the universe. I don't know. So we can learn if there's some kind of organic material there that probably this theory can be supported in any way. That's the thing that I think, well, maybe we are going to learn a lot of things about the movement of these asteroids through the space. But yes, from a biology point of view, probably this would be the most remarkable thing.
Awesome, thank you. Next up, do you think that life originated on Earth, or was it brought to Earth by an asteroid?
Okay. So we are going back to Panspermia. Why not both things? Let's say why the basic chemical organization came from an asteroid, and here on Earth found specific situations to develop. Why not both? Yeah, while I was during my years of studies, I started thinking that life was originated in below the surface, in oceans, in these thermal springs that with thermal. I don't know if the word is spring, probably not. The thermal places beneath the water surface, where they can have this energy coming from the volcanic activity on the crust of Earth. They will have this light coming also from chemical reactions. They will have nutrients. Well, that was my first impression. But after reading more things and after discoveries like the one of the ethanolamine, I'm more about a mixture of things. I think it's a very important and a very interesting thing just to try to find the origin of life. But they are still theories at this moment. I don't know if taking further steps will give us some other solutions or some other, maybe even more theories. But at this moment, I will put my money on a mixture of things.
Nice, cool. Have you seen any competition between different types of bacteria in these environments, and any potential for antibiotics?
Yeah, that's a very good question. Yeah, also in our lab we have a guy that is finishing his PhD that works with these kind of things, especially in the Lost Hammer Spring and in the Arctic. He's trying to find if the microbial communities there can create different antibiotics that we already know, just to be employed on different things that has a human importance. And one of the coolest things that he has found is one of the strains of one bacteria there can create antibiotics that works in the culturing of wheat, of cannabis here, and they are applying these things to the leaves, and these cultures prevent the leaves to be infected by fungi. So yeah, there are many other things. I would like to say that antibiotics are not always for competitions against microbes. They are also kind of signaling molecules. Like, okay, you will have to one group of a community of microbial species or whatever, just use these microbes to signal the others to be ready for anything, ready for a nutrient input, ready for a nutrient scarcity, ready for competition, but they are not always they are not always intended to fight against other strains, other species or whatever.
I see. Thanks. All right. I think this will be our last question. What results from your research in studying extremophilic microorganisms have surprised you most?
Yeah, well, to be honest, the most surprising ones has been the last ones, that the one that I talked about, the Nunatax one, because this study started as a microbial ecology study, and we did a dedicated sampling trying to study different situations on one mountain, like different heights, different depths, different things. And I was expecting to have a higher diversity of microbes in soils than in rocks, for example. So because I know that these islands where we sampled the things are not so cold, not so dry. They have much more water, especially in comparison to other places like Atacama, where we have found the the rock inhabiting microbes, and also the ones in the dry valleys where you can find this rock that I have shown you. But in this place where the temperature is not so cold and the water is present, I would expect they will be more comfortable on this soil than in rocks, because rocks, it's a very extreme environment. And also the ones that we are having now with the Mars chamber experiment are also surprising me. I cannot say much about them because we are just receiving the latest results. But yeah, because we are trying to find the changes in lipids that are the ones that forms the membranes, that are supposed to be the most resistant biomolecules that you can find, and also the DNA and some proteins. And once would expect that the DNA, that is the more label one that degrades the most or faster than the other ones, would be the molecule that's going to be more affected. And the preliminary results show the opposite. So we have to think about why this is happening, and so it's kind of surprising, but also it's kind of cool. When you work on science, you are looking for unexpected things to have fun with them. Normally, it's difficult to explain what you have found, but it's exciting also.
Yeah, that's wonderful. All right. Thank you so much, Miguel, for your talk this evening. Everyone who is watching on Zoom or watching along on YouTube or in the comments, please give your thanks to Miguel. We will make sure that it gets to him. Yeah, and have a wonderful evening, everyone. Please watch our social media channels for our next event. Have a